Hi,
Hey, how are you?
I'm good. How are you?
I'm good. What's new?
Um, well, I have the last two practice tests that I took AMC full length one and AAMC full length two. I kind of like got the same score on both of them, so I've been like slightly discouraged, but also not too discouraged. But I guess I just need to figure out like a different study regimen for over the next few weeks, because I mean. I'm still applying this cycle and I was like debating like giving myself an extra 2 weeks just to see like what content gaps I'm missing because I feel like at this point it might be content gaps. So I was
Okay.
wondering if you had any suggestions for like studying content over the next 2 weeks, like if I should take a break from like taking a practice test and just focus on filling in the gaps that I have and
Okay.
then
So let me first just input your full-length AMC full-length 2 score into the spreadsheet. So when did you take it?
I took it on Monday because on Sunday I wasn't feeling that well. Like, I don't know what happened. I was just tired.
So the night 19th.
Yeah. And it's the exact same score as full length one.
Same breakdown too?
Yeah.
Okay. So 124, 124, 124, 123.
hmm
Okay. And were you able to blind review and review it?
Yeah, I didn't finish it, but I did the psych soc section. I started with that section first just because I mean, honestly, I really haven't put too much time into psych soc yet, so I figured like it was something that I was stronger in undergrad and I was pretty good at. So I was like, let me start putting time into this and like starting to learn those what do you call it? The definitions and stuff like that again. So this is my psych soc board so far.
Mhm.
LOL. So I did the psych soc section yesterday and then I started doing biochem today. Um and then I'll do like cars and chem phys.
Yeah. So yeah, the reason I ask is because your blind review would be able to be a good indicator of whether your issues are with, the content or not. Meaning like if you got a lot more, like let's say you get like more than half of the questions that you do during your blind review correct, it seems like it's less so a content thing and probably more so just like, the time pressure. But if you're getting, after you do your blind review, if you're getting, let's say, less than half correct, then maybe it's content stuff. So you could also kind of like show me the types of questions that you got wrong, and I can kind of see like, okay, if this is like mostly content stuff or not.
Okay, yeah. Yeah, I'll show you. I guess I could show you for my what do you call it? Biochem section. Let me share my screen.
Yeah, and did I have you do the Jack Weston content diagnostic when we first met?
No, I didn't no, we didn't do that one.
Oh, okay, okay. That could be all right. That could be a good next step to do. I'm wondering why I didn't have you do that. Um, yeah,
I don't know why, but I like to start backwards, I guess. But I noticed I got like a good amount of like the discretes wrong in this exam.
I see, I see. So
Yeah.
for this one, um, like the difference between like a native gel, SDS, like denaturing gel, and reducing gel.
Yeah, so like the SDS gel, doesn't that have like a negative charge?
Okay, this could be a good thing to go over real quick. So you're basically going to have, say, these are the three types of gels. Now, let's say that we have, I'm going to just make up a protein. So let's say that we have a protein with an alpha subunit that is attached through a disulfide bond to, let's say, a beta subunit. And then let's say this is like some hydroxyl group of like some amino acid in the alpha subunit. And a beta subunit can also have this hydroxyl group. So this can be an intermolecular bond, like a hydrogen bond.
Hmm.
Let's say that the beta subunit is then attached, or wait, I'm gonna make an adjustment here. So this is another alpha one, let's say, and then it's going to be disulfide attached to another beta one, which is going to be, you know, it could be attached to the other beta one through hydrogen bonding as well. Okay. Now this is called well, so alpha, beta, each are subunits. So this has a quaternary structure, and this is actually an example of a homodimer, which mean the homo means the same, the
Hmm.
dimer means two. Like overall, it's a tetramer, meaning four subunits in total, but but it can exist as a homodimer where you have the alpha subunits attached to the beta subunits, as well as, each alpha attached to another alpha and each beta attached to another beta. Now, let's say that the weight of an alpha subunit is, let's just say, 50 kilodaltons. And then let's say that the weight of a beta subunit, let's say, is 60 sorry, 60 kilodaltons. Okay. Now, this entire structure would weigh how much?
It would be 120 plus 100, so that's 220 kilodaltons.
Okay. 220 kilodaltons. And now this will be the native gel. This will be the SDS gel, which in other words just means denaturing. And then this will be the reducing gel. So now what is meant by denaturing?
So like the disulfide bridges would I guess they would denature. I guess the protein, it wouldn't be a tetramer anymore. I think they would all split up into their own subunits.
But specifically tell me about what denaturing is.
It's the breaking of the bonds that is are holding the subunits together. So those hydroxyl bonds would break and the disulfide bridges would break as well.
Okay. So now let's take a step back. Let's think about protein folding in general. So your primary structure, what would that like be composed of?
Um, the primary structure is just like the peptide bonds between each amino acid.
Okay. And what functional group would that be?
Um, the amine group with the carboxyl group,
So that will be called something.
like the whole like the bond itself t together would be called something. Oh yeah, the amide.
Yes. Good. So yeah, you want to be able to take like being able to see things interdisciplinarily. So yeah, on one hand, we call it a peptide bond, but on the other hand, you also want to know that the functional group for a peptide bond, it's exactly what you described, which is a carbonyl attached to an amine. I'll just say this is like R R. So, but you also want to notice that this is an amide or amide, and that grants it so the nitrogen of the amide here has a lone pair, right?
Mhm.
Like nitrogen has five valence electrons, and this nitrogen has three bonds. So there's a lone pair. This lone pair can go here to make a double bond there, and this lone pair can go up here to well, make the oxygen negatively charged. So this would look like O minus, double bonded NH, R, like that, right? Through resonance. So this amide bond that makes the peptide bond, it has partial double bond character. And we know double bonds are stronger than single bonds.
Yeah.
We also know resonance like makes a molecule more stable. So this is a very, very stable and strong bond. Okay. Like, and making it from two different amino acids, right? I mean, they could be the same, I guess, but two different amino acids, making that is going to be through dehydration synthesis. And breaking it would be what?
Hydrolysis.
Very good. So that's our primary structure, and it can only be broken through like a really, really strong acid or base. So we're typically not going to see that it's breaking. Oh, well, I said typically broken with an acid or base through some type of enzyme like a protease. That's the only time that we're going to see this bond break. But when it comes to the gels, we're not going to ever use anything as strong enough to break the peptide bond. So that primary structure is going to be intact. Now, your secondary structure can what can you tell me about that?
So that's like the alpha helixes and the beta pleated sheets, and they are stabilized by hydrogen bonding.
Okay. Now, for any amino acid, right, there's an R group, right, that dictates what that amino acid is.
Hmm.
So now your secondary structure is gonna involve hydrogen bonds. So, and by the way, so like you have like your intramolecular bonds, which we draw like as a line. So like the bonds that I drew for this molecule, like it's a line, right? So that's we're gonna make that be like a covalent bond. Um, but then you have your intermolecular bonds, which are never drawn as lines. And you can see here, I kind of use like these kind of dashes to draw the bonds between the OH groups.
Hmm.
Whereas for the disulfide bond, I use like a line. So the line again indicates covalent, and the dashes indicate non-covalent, like intermolecular. So that could include your van der Waals or London dispersion forces, which will always exist. That will never not exist. Um, Electrostatic interactions you can have. You could have dipole interactions. And then you can have hydrogen bonding, and out of all the intermolecular bonds, hydrogen bonding is definitely the strongest, but it's still super weak compared to a covalent bond. Okay. So secondary structure does involve hydrogen bonds. However, it'll be hydrogen bonds between the backbone, the peptide backbone. It's not going to be hydrogen bonding between the R groups. Okay. So in other words, like, so this is like a drawing of a peptide. So if you imagine another one here, you could have, I'll use red, these dashes between the hydrogen of the, because you know, hydrogen bonding requires either FO or N
Hmm.
hydrogen, right? So this
Yes.
nitrogen with an H can be a hydrogen bond donor to the carbonyl oxygen. And so there's a hydrogen bond there. Um, and that's what's meant by between the backbone. It's not between the R groups, okay? So that makes your secondary structure. Your tertiary structure is where everything comes in. So that's when you have your van der Waals, aka London dispersion. You have your Dipole uh bonds, your or interactions. You have like your electrostatic interactions, which could be like something like, a carboxylate ion and then like an NH3 plus, amine. You could have electrostatic interactions happen there. And then you can also have, so I guess I'll write like van der Waals, comma, like dipole interactions, electrostatic, and hydrogen bonds, except this time between the R groups.
Yeah.
If you had a like a ser I'm pretty sure this is a serine here and another serine here, you could have hydrogen bonding between the R groups of each of these. R group being the OH for serine,
Hmm.
right? And that is involved in tertiary structure, but the predominant interaction that causes tertiary structure is hydrophobic interactions.
Yeah.
So, yeah.
Oh, no,
So, yeah,
I was gonna say because that makes it like fold in on itself.
exactly, exactly. Um, and that's like, it's not even really like a type of bond per se. Like, van der Waals is a type of bond or interaction. Dipole, electrostatic, hydrogen bonds are all types of bonds. Hydrophobic interactions are less so bonds and more so the fact that if you have a protein, and like tertiary is also when we're starting to deal with uh three-dimensional like proteins. Let's say that this green region and this green region are hydrophobic and, inside of the cell, it's mostly water, so it's mostly a aqueous environment. So there's a when the water attempts to solvate the hydrophobic part, it creates a solvation shell, which is like a very ordered cage around it. And if it's very ordered, it's low entropy. And the universe favors entropy or disorder. So it has to pay what's called an entropic penalty. And to pay it, it has to fold, as you said, fold in on itself so that the hydrophobic parts will be sequestered on the interior, making it, you know, more stable because the hydrophobic parts are trapped on the inside. And that's folding. And now denaturing is just, so right here, to denature something, it's to unfold it. Okay. So, okay. So like if this gel is running, in this direction, um, and like in a gel, like it's made of like these tiny pores. So the lighter something is, the further it can travel through that gel. So this 20, oh, I wrote 22, 220. So the native gel is not going to do anything to that protein. Like it'll keep it the way it is. I mean, like, yeah, you can add like a negative charge to it just so it can be receptive to the electric field and cause it to move. But it's not going to move very far. Let's say it moves like down here. And this is a location where, let's say this is the 220. kilodalton value, and it'll just be there. Now, when we move on to SDS, in other words, denaturing, you're going to break apart all of the predominantly like the non-covalent bonds. Like you're going to break apart the hydrogen bonds, the dipole interactions, the hydrophobic interactions, the van der Waals forces, but you are not going to break any of the covalent bonds. So you're not going to break the peptide bond, but you're also not going to break the disulfide bonds because those are also covalent interactions, right?
Oh, okay. Yeah.
So if we denatured it, we would break which bonds on this?
The OH bonds.
These, right? So you would end up with two alpha beta uh, fragments, right?
Hmm.
And how much would each of these weigh?
110.
Mm-hmm. So around here would be like 110. And since we have like, two of them, like you're still gonna have one band, but it might be a thicker band.
Gotcha.
Okay. And now for the reducing one, and also I should have said this. So like if you have, you know, um, so this thing that I just drew here, this is just a way for me to draw like a squiggle to for me to draw the amino acid with just the R group. Okay. So I'm just draw. So like, what amino acid would this be with this R group?
Cysteine.
Very good. So if I had two cysteines, in order for me to make it, a disulfide bond, what type of chemical reaction is this?
Um, I would say it's a no, it's not a combination. Is it reducing?
So,
Oxidation?
yeah, yeah. So, for oxidation, um, in biochem, I think maybe we have talked about this before. I'm not sure, but um, because in my PDFs folder, I have a PDF called Biochem Redox.
Hmm.
Kind of gives you the generic kind of thing that happens to functional groups when you oxidize or reduce them. So because when we think of redox, we think of like Gen Chem where like an oxidation state changes, like of a metal or something, but that's not very relevant in like biochem. So you can
Yeah.
think of oxidation as either loss of hydrogens, gain of oxygens, or gain of bonds to already existing oxygens. So this is going to be an oxidation, which then would make sense that going from the disulfide to breaking it would be reduction, right?
Yeah.
So for the reducing gel, we are going to be breaking the disulfide bonds, right? In addition to the denaturing.
Okay, that makes sense. Does the reducing gel have like a negative charge?
When you say the gel,
Yeah. I mean, well, I guess I'm just confused because I thought that there was a gel that had a negative charge to it, but I guess maybe I'm wrong.
well, in gel um electrophoresis, you're going to produce that um like it'll be there will be an electric field that's produced on the gel itself. And typically you're going to have, well, I guess you're thinking about when you have the SDS gel, which can impart like a negative charge on the protein that makes it receptive to the electric field inside of the gel,
Mhm.
or maybe not inside of the gel, but across the gel. So that is something that your SDS will be able um, will be able to do for the protein. It does make it have a negative charge, which makes it able to move, um, along the gel itself. Um, so,
Yeah, I guess, but that's not like the main point of the SDS show, I guess.
mhm.
Okay, yeah, maybe I was just paying attention to the wrong thing.
Oh no, no, no. That's I guess still an important thing. Because I guess, well, yeah. So like in order for anything to move in the gel, yeah, it's gonna have to have um a charge. And typically like a gel is used with well, I was gonna say like DNA, which by itself has its own um negative charge. Um and so for a protein, like any given protein is gonna have some positive charge, some negative charge, so a protein can still move in native. Um but yeah, yeah. So um you could have the um yeah, so I guess that is a part of it, but yeah, the when you see it compared to like native and stuff, usually you're gonna see that it's just gonna involve the denaturing aspect of it. Now, okay, so now if we have a reducing environment, we are, in addition to denaturing, breaking the disulfide bonds. So what type of bands should we see?
So then I feel like that's when we would see the separation between the bands. So we'll see two of the alpha bands and two of the beta bands. But I
Uh-huh.
guess I think it
Yep.
will just be two like thick bands because
Mm-hmm. Yep. So it could be something like that and something like that, right?
yeah. Okay, I guess doesn't have anything to do with this question, but I think I just remember doing a practice question where they were like asking like about like the molecular weight at like the bands.
Mhm.
And see how it's like at 50 kilodaltons, it's just like one band, but if they were to ask like how many kilodaltons is the band at 50, would it be 100?
Tell me uh tell me about the example that you're talking about.
Like I think it was like asking about like the weight of like the amino acids at like a band or something like that. And I think is it because it's like two alpha subunits, would it be 50 kilodaltons at, I mean 100 kilodaltons at the 50 band because there's two of them?
Oh, well, since we're in the reducing gel, since we're breaking up, so like we're gonna end up with two of the alpha and two of the beta, and each of the alpha weighs 50. So and each of the beta weighs 60. So they'll still reach the 60 kilodaltons in beta, sorry, and 50 kilodalton value rather
Mhm.
than be up at 100 and 120 respectively. because we have the two like this, right? And each of them, like basically, like yeah, each of them will just like each of the ones for the beta will reach that 60 kilodalton level, and each of the alpha will reach the 50 kilodalton level. So they would only be like the 110 or something like, yeah, if you had something like that.
Okay, I see what you're saying. Okay, well, yeah, I feel like I get it.
Okay. So let's see for this question. So they're saying a homodimeric protein was found to migrate through SDS gel with a mobility that matched that of a 45 kilodalton standard. Oh, and then when it comes to these gels, you're gonna see something like, well, like because like how do we know that this level here is 120, right? So like you're gonna see some test band that could be like, at 220, at like 110, or like at like 60 and stuff. Like a test standard is how we know what those numerical values are. So they're telling us here it's a 45 kilodalton standard. Okay. And then what change for the experiment will increase the chances of observing the mobility for the 22.5 kilodalton monomer. So so they're telling us that the homodimer weighs 45, and a homodimer means that you have two of the same subunit bound to each other. So if they're the same, then it would make sense that each of them would be 22.5 kilodaltons, right? Because that'll add up to the 45.
Yeah, that makes sense. Although,
Yep, yep. And so like, yeah, increase the gel running time. That won't change that. It can make like the resolution better. Like if our resolution was like really like a thick faded kind of, you know, band, running it longer can help like make it like thinner or thicker at like the correct location instead of like a kind of a I'm trying to think of the word. It's like when you um s smudge. Yeah, like it looks like a smudge, kind of like that.
hmm
So okay. Um, Using a higher voltage won't change it. Removing the SDS. Now, that's a good thing to talk about because since proteins fold on their own, like spontaneously, and since denaturing, like SDS denatures it, basically, but like there's also like beta mercaptoethanol. Um,
hmm
they basically disrupt at the secondary structure level, which can then kind of uh, mess with the tertiary structure and stuff. But the thing is, since folding happens spontaneously, if you remove the SDS or whatever is causing denaturing, the protein will spontaneously fold back to its native structure. But if you reduce it and break the disulfide bonds, and then you remove the reducing agent, it's not gonna reform the disulfide bonds because in order for it to form the disulfide bonds, it had to be oxidized, right?
hmm
So so you could reverse the denaturing of a protein by removing the denaturing agent, but you cannot reverse the breaking of disulfide bonds by simply removing the reducing agent. You would have to add an oxidizing agent. So here, the only choice that will actually work is by adding a reducing agent, because then that will break apart the disulfide bonds. And we know that there must be disulfide bonds here because when they used the SDS and they got the 45 kilodalton one, and they're telling us it's a homodimer, we know that it could be broken down even further.
Yes.
So that's why it should be B.
Yeah, that makes more sense. I'm like, where did I put
Mm-hmm.
a D?
Yep. If you want, you could take a screenshot of this.
Yeah, I just didn't know this one. I just did not know it. I didn't study the reproductive system yet. Um,
Oh yeah, I think there's like a mnemonic for that that's like 7 Up.
7 Up. Let's write that down real quick.
Yeah, let me see. It's seminiferous. Here, I'll just put this in the chat.
Oh, okay.
Yeah, I'm pretty sure that could answer it. Well, they're asking become motile and capable of um fertilization. So that's where, because like there's in meiosis, there's like the primary oocytes or like the secondary. There's like oogonia, and then like, oh no, well that's for females, but like spermatogonia, and then something like sperm. It's like primary spermatocytes, secondary spermatocytes, something like that. So like yeah, so like my something with meiosis.
I'll look it up and I'll figure that out because I feel like I don't have too much trouble with the
Sure.
discretes. I feel like most of the time I get the discretes wrong if I just don't know it. I also just didn't know this. I have to study the mesoderm and stuff like that.
Oh yeah, that could be kind of annoying. I have a good um image for that here. For the most part, it's like kind of straightforward, but then there's some oops. Then there's some that are just kind of like completely out of left field. Here, I put it in the chat. Like we know that like the ectoderm is going to be things that are on the outside, so like your skin, your like nervous system and stuff. Mesoderm is stuff on the inside, and endo. Well, I guess mesoderm is like stuff that's like in the middle, I guess, and then your endoderm is going to be like the epithelial layer of like your GI tract and stuff.
Mm-hmm. Fun.
Hmm.
And then I redid this. I started with this passage just because I got all of them wrong, but I was able to get this question right second time around. Um, but I still was not necessarily understanding. So I was wondering if we could go over this passage, um, if we have time.
Okay, so let's have you start by reading it and writing and telling me if there's buzzwords that you see and also what proportionalities we can write.
Gotcha. So Ebola virus causes a rapidly progressing and often fatal hemorrhagic disease that currently has no effective treatment. So I guess buzzword is Ebola virus and fatal hemorrhagic disease.
Uh,
Um,
yeah. So like that's like the kind like the passages will always start off with something like this to be like, hey, we're still talking about clinical stuff, but then the rest of it'll just be like the non-clinical like science stuff that we're supposed to know.
mhm.
Oh, yeah.
Um, Ebola particles consist of a nucleocapsid encased in a membrane that contains transmembrane viral glycoprotein, EGP. So I think that's a buzzword because it comes back up again, like EGP, which is the um, transmembrane viral glycoprotein for Ebola,
Okay.
I guess.
Hmm.
Okay. This virus has a single-stranded negative sense RNA genome that is used as a template for the mRNAs produced by a virally encoded RNA-dependent RNA polymerase. Um, I feel like single-stranded negative sense RNA genome is important.
What
Um,
does it mean by negative sense?
I think it that means it's going in the opposite direction of
So,
what.
so the positive sense is going to be like, okay, like if we talk about DNA, I know it says RNA, but if we're talking about DNA for a sec, so DNA can have a coding strand and a template strand, right?
Mhm.
Like if we had like A, C, G, T, A, like in the 5 to 3 direction, in the 3 to 5 direction, this will have to be T, G, C, A, T, right?
Yeah.
And let's say this is the coding strand, like this is the strand that codes for the gene. This could be considered the template strand because it's complementary to the coding strand. So when RNA gets transcribed, it's going to get transcribed by using the template strand. So the template, like meaning it'll be complementary to the template strand. So this T could be A, C, G, oops, U, and then A. And so this is the coding, this is the template, and then this is like the coding, mRNA. And you can see how it has the same sequence as the coding strand, but it can only do that by using the template strand, which must be complementary to both, right?
Oh, okay. Yeah, it's like right in the middle.
Yep. So when they're saying negative sense, so like also this is positive sense, this is negative sense. So when they say negative sense, they mean the template. So this Ebola virus has the negative sense RNA genome, and they even say it's used as a template for the mRNAs produced by virally encoded RNA-dependent RNA polymerase. So if a virus is a negative sense virus, the mRNA that it has isn't this mRNA. It's actually the RNA version of the template. So that's going to be U, uh, G, C, A, U, um, in 3 to 5. So this is going to be like the negative sense RNA genome, and it itself is not coding for the gene. That's why it needs to bring its own RNA polymerase, which can then use this negative sense template RNA to make the mRNA that's coding, which is complementary. So complementary to the U will be the T, and then this will be the C, this will be the G, this will be U, and then this will be, oh wait, I put a T there. Sorry, this should be A and then A. So the Ebola virus can contain the template strand, and to make the coding strand, it needs its own RNA polymerase. Okay.
Gotcha. So,
Mhm.
okay. Okay, I'm just trying to make sure that I understand. I guess so it's just kind of like moving backwards in a sense.
Um, yeah, let's see. I can get a
Because I'm like, okay, because like the mRNA, like they're just coding for like proteins, correct?
uh-huh.
Okay. Okay. So
Um,
they don't really have to go from like the RNA to DNA.
for the yeah, for this, since it's already
Okay.
starting with RNA, but I put another image in the chat, and if you could, yeah, if you can pull that up.
Oops. Can you see it or oh, hold on, let me see. Can you see the image?
Um, no, I think you're just sharing like the tab, so I can
Oh, my bad, my bad. Hold on.
share the screen.
I just get to share. You're making me pick one.
Yeah, there should be one that just says screen, and then it'll just share whatever's on the screen.
Apologize. You know, I'll just do this entire screen. There it is.
Yep.
Cool.
There we go. Yeah, so here we go. So we see we have whoops. So first let's look at the one on the right, the negative, I mean they just wrote minus, but negative sense single strand RNA virus. So we have the virus here, and it injects its genome into the um, cell here, and then the mRNA that, or the RNA that it has can be either like replicated or transcribed into this, which will be the actual coding strand.
Mhm.
And then that, like the blue, okay, so the blue is the coding strand, and then we can see like that, like the mRNA coding strand. And then we can see that the coding strand just gets translated into the proteins, right? Whereas
Yeah.
plus sense, it can just inject its RNA, which can directly be translated because this RNA is already the coding strand.
Oh, okay. That makes sense.
Yep. So a plus sense or positive sense single-stranded RNA virus does not need to bring its own polymerase because the RNA that it has in its genome is already the coding, like it already codes for the protein. Whereas this one on the right, it's the template, so it needs to transcribe that um, to make the mRNA that can then be translated through the protein's, ribosomes. Okay,
Cool. Thank you.
good. Okay.
Okay. So the entry of an Ebola virus nucleocapsid into the host cell cytoplasm starts with the fusion of the viral membrane with a host cell membrane, a process facilitated by EGP. So, I guess we can make like a proportion for this because EGP causes fusion of the viral membrane and host cell membrane.
Okay, we could say the entry of a EBOV nucleocapsid into the host cell cytoplasm starts with the fusion of a viral membrane with a host cell membrane facilitated by EGP. So maybe I would say like, okay, EGP is proportional to um, EBOV entry. Okay.
Recognizing the similarities between how EGP and the glycoproteins of related viruses are processed during viral infection, as well as how inhibitors of endosomal acidification block EBOV infection. Researchers hypothesized that acid-dependent protease trigger the conformational changes in EGP that are necessary for fusion. So, um, I guess well, first it needs the acid-dependent proteases in order for it to fuse. In order for it to EGP to be activated, I guess.
Yeah, let's see. So they're saying that they looked at similarities between how EGP works alongside other or glycoproteins of related viruses, and then also how inhibitors of acidification can block EBOV infection, led researchers to hypothesize that acid dependent proteases can trigger the conformational changes in EGP that let it fuse, and fuse we equate to like entry. So we could say that, let's see, we can say that this could be 1 over pH, meaning the lower the pH, or wait. Well, okay. The lower the pH, the more likely this EGP can enter into the host cell. Maybe I should say, here, I'm just gonna say EBOV is proportional to acid-dependent proteases. So 1 over pH, I'll put like a semicolon, proteases. Yeah.
The infectivity of VSV EGP vesicular stomitis virus particles engineered to contain EGP instead of VSV glycoprotein in the viral envelope was reduced more than 99-fold by inhibitors of the mammalian proteases, CAT-B and CAT-L.
Okay.
Um, so now they're doing like an experiment on VSV EGP um, to to look at how it works with the inhibitor,
Mm-hmm. And what do the inhibitors inhibit?
the proteases,
Which ones?
the mammalian proteases.
All right, what do they call it?
Oh, cat B and cat L.
Hmm.
Sorry.
So, okay. So let's see. So the infectivity of VSV EGP, and then they tell us VSV is just some other virus that they engineered to contain EGP instead of VSV, and EGP is a type of glycoprotein. The infectivity dropped when they inhibited cat B and cat L. So I'll say this could be inversely proportional to cat B, also inversely proportional to cat L, because, well, wait, no. Cat B and cat L increase the infectivity, but inhibiting cat B and cat L can decrease the infectivity. So I'll just say it's proportional to cat B and cat L, meaning if you have more cat B or if you have more cat L, you're going to have more infectivity.
Gotcha. In a separate experiment using a strain of EBOV, Cat B inhibitors reduced the number of EBOV particles produced in cell culture tenfold. So yeah, I think yeah, you just said that. And figure one shows how the introduction of certain genes into two mutant mouse cell lines affected the subsequent infection of these cells by VSV EGP.
Okay, so this part saying particularly for inhibiting cat B, right?
Mm-hmm.
Okay. Um, all right. So, and then they did an experiment where they introduced certain genes into the mutants, and then they looked at the infectivity of these cells. So, okay, we have effect of cat B on cat or cat L on both or both on VSV EGP infectivity. So, what yeah, how would you analyze this chart?
I feel like this I was not understanding it when I first seen this graph, but I think I get it. Like I feel like the wild type like okay they didn't put any extra genes in that one. Um and then they have the two separated I guess let me use my mouse. They have like the two separated by the cat B inhibition and then like L is not inhibited. So with cat B that's inside this gene, it looks like I'm kind of confused why it's if it's inhibited, why is it like
Got it.
having
So the top here indicates the genome of the culture or the host here. And so, okay, with the wild type, that means it has the gene for cat B and cat L, and we have some value that's like around 26 or something percent of cells infected. And then when we have the cat B knocked out, but the cat L like is still there, the bottom the x-axis here indicates the introduced genes. So if you don't have the cat B in the genotype, but you introduce cat B, it's ultimately going to be having both cat L and cat B then, right?
yeah.
So now I think when they mean bacterial, I think they might be talking about like the host cell, I think. So, I mean, I could be wrong, but I think that, or maybe it'll be better to compare it to this part, this one here, because this one means that we have cat L, which we already have in the genotype, but we don't have cat B, right?
Yeah.
So we have a very tiny level of infectivity, right?
Okay, so I guess this is what it's saying that in order for cat L to infect, it needs cat B as well.
Might be. So let's see, right? Because if we don't have cat B, but we have cat L, we have almost no infectivity. But
Mm.
if we have cat L and cat B, which is this, we have boom, it's a super high amount of infectivity, right?
Oh, okay. Yeah, that makes sense. Yeah.
So we could say that, yeah, cat B produces a very high amount of infectivity. Um, and now when we look at the next genotype, it's lacking both cat L and cat B.
Hmm.
And so if we have cat B added to it, compared, and again, I think the bacterial is the host cell, like, you know, without, with the genotype here. So, it's like next to nothing. When we add cat B, it goes up a tiny bit. When we add cat L, and by the way, so when we add cat B, it means we have cat B, but we don't have cat L. And then this one for cat L, it means we don't have cat B, and we originally didn't have cat L, but now we added some, and it doesn't
Yeah.
seem like it does much, right? And
Yeah.
then here we have cat B and cat L, and it seems like it has the largest effect, right? So what I would do here is now I would just write like a greater than hierarchy. So I would write in terms of like infectivity, the highest, so like the options are no cat B or cat L, just cat L, just cat B, and then cat L and cat B. What do you think
Gotcha.
would be the highest?
So the highest is cat B in terms of infectivity.
So out of the options of cat B, all right, so like cat B, cat L, neither, cat B and cat L,
Yeah.
what would be the highest infectivity?
Yeah, that would be cat B, right?
So when I say cat B here, I mean cat B only.
Oh, I see what you're saying. Okay. The highest definitely would be cat B plus cat L.
Yep. So cat B plus L. All right, next.
And then it would be cat B.
Next.
And then it would be cat L and then neither.
Good. So we could see that cat B, if we had to choose between cat L and cat B is definitely better, but it's the best is always going to be the combination of cat B and cat L.
Hmm.
Hmm.
Yeah, I feel like I know what I was missing before. I feel like I kept forgetting that cat L, like this little detail where cat L is also part of this. I feel like I wasn't like factoring that in
Yeah,
when I first looked at.
so when they have like, a minus dash minus kind of thing,
Yeah.
what they mean by that is that the gene, and also since they're saying the genotype, that's what these mean. They mean like, so it means that the cat B one was knocked out, cat L was, maintained. And then the introduced genes is them like, kind of like just introducing it or adding it to it. And then we just want to know like, yeah, what the effect of these are on infectivity. And then also when we read the passage, we wanted to see that EGP is simply something that helps this Ebola virus to enter into host cells, and that
Mhm.
proteases can enhance that, and the proteases are activated in in acidic conditions, because you can see here they even wrote like if you inhibit, like, sure, here we wrote that you could inhibit the proteases of cat B and cat L, but here they tell us you could also inhibit by inhibiting whatever causes acidification. So that could be another thing that they could ask.
Yes.
Mhm.
Um, yeah, so for this question, I at first I thought I was like confused, but then I like when I went back, I saw that it was like in the passage that's like um endosomal acidification.
Mm. Oh yeah, here, right?
Mhm.
Yeah, what is an endosome?
Um, I honestly I can't remember. I feel like aren't they like those little things that don't they create like little pockets in the cell membrane kind of?
Yeah, yeah. It doesn't have to be in the cell membrane, but inside of the cell, it's basically like a secretory vesicle that can bud off from something and be like its own, it's almost like a little bubble. Um, let me throw a picture in here for you. So, okay, I'm putting an image in the chat. You don't have to share it, but it's basically like like its own like kind of little cell membrane kind of thing.
Hmm.
Yeah.
Oh, it's cute. Sorry, my bad.
hmm
I like the picture.
hmm
Yeah, I was thinking about endocytosis. That's why I said membrane, but you're right. It could be anywhere.
Oh yeah, yeah. So the endocytosis is when like cyt refers to cell, endo means inside, exo means outside. So if it's exocytosis, it you know like leaves the cell, endo takes it in the cell. Um,
Mhm.
but like oh, I guess I wrote escape wrong, but basically the endosome is like itself, like, it's like its own little cell inside of a cell, like it has something inside of its membrane. Um, and this is something you see a lot in viruses, like this. I'll put this in the chat as well. But basically, like how viruses, enter into our cells is that they have these endosomes that can um yeah can enter that can fuse with our membranes and enter into our cells.
Compounds.
Mhm.
Okay. So for question 40, it says, based on the passage, is CAT-L expression sufficient for VSV-EGP infection of mouse cell lines presented in figure 1? Um, answers.
um okay, so what is this asking us?
It's asking if all we need is CAT-L in order to
Okay.
have for infectivity, I guess.
Okay.
Um, at first I put yes, but I guess I wasn't looking at the whole thing.
Yep, so show me on figure 1 where the bars would be for just having cat L.
Just having CAT-L would be here.
Okay, anything else?
Here,
Okay, anything else?
and I mean, well, this has cat B in it, so not that one. And then Here.
All right. So all those values are pretty low, right?
Mm-hmm.
So do you think cat L expression sufficient?
No, no.
Yeah.
So that's the first thing because it's not.
So get rid of these.
I'm so confused by this wording. It's like no, because VSV EGP does not infect cat B minus cells expressing cat L better than it infects cat B minus cells not expressing cat L. Um,
So show me where cat B minus cells express cat L.
that'll be here and here,
Okay.
and then okay. And then no, because VSV EGP infects cells expressing both cat B and cat L better than it infects cells expressing cat B but not cat L.
Sorry, so for the previous one, you said that the cat B minus cells expressing cat L is where?
Here, um, and here.
Okay. And then it says compared to cat B minus cells that don't express cat L, where would those be?
Here and here.
Okay. Cat B minus cells not expressing cat L. So that could
Oh,
be,
yeah, like
yep.
that's true.
Mm-hmm.
Like the basically the same.
Mm-hmm. Yep. And because they're telling us also here that if its values are less than 3% of the total cells infected, so we know here is 5, and then I guess we can pretty much reasonably say that, this is 3%, and so if it's less than 3%, it's too low, and so we can say that these three have pretty much the same levels, right?
Mm-hmm.
And then D would say that expression that the cells have both cat B and cat L is more infects more so than things that have cat B but not cat L.
And that's not true.
Uh-huh. Because it says here, so this is cat B and cat L, and well, actually, I think the expressing part, let's see, not express or expressing both cat L and cat B better than it expresses cells exposing cat B but not cat L.
Oh, never mind.
Okay. So I guess we don't have an example of, like, I was thinking maybe wild type, but I don't think we have an example of cells expressing both cat B and cat L.
Oh, isn't this one here?
Yeah, I was trying to see, like, when they say expressing, like, I think that means here, whereas this stuff down here is like them adding it,
Oh, I see what you're saying.
expressing both cat B and cat L better, that it infects cells expressing cat B, but not cat L. That's okay. So like that's also hard because if we said that cat B and cat L is this, meaning it has cat L but not in its genotype, but not cat B in its genotype, we can add cat B. And then this would be cat B and cat L both. And then
Hmm.
compared to just cat B over here. Yeah, so I think D. Yeah, so I think D is something that we can't ascertain. And I think it also doesn't answer the question of is cat L expression sufficient? So yeah, so I think when they say like expressing, they are talking about the genotypes.
Okay, that makes sense. I guess that's why they mainly asked about cat L because there's no cat B expression.
Yeah. So I think while D, even if we say that expressing could also indicate this, we still are like wouldn't be answering the question of is cat L expression significant, right? Because if we said cat L was not sufficient, um, and then we say that the cells expressing both cat B and cat L infects more than cells having just cat B, like that would give us information about cat B, but not really about cat L.
Yes. I don't know, I felt like this was so confusing when I was doing it.
Mhm.
Um, like I feel like I got this one right the second time.
Okay.
Got that. Yeah, I figured out. I was like, oh, it's a protease. Um, okay, yeah, I was confused by this one. So the precursor of EGP is translated from a transcript that has had one non-templated nucleotide added to the open reading frame. This change does not create or eliminate a stop codon. Compared with the protein SGP, which is produced from the unedited transcript, EGP most likely has the same primary. Um, so yeah, I was confused because I was like, oh, SGP is not in the passage. I was like trying to see if I could figure out like if they gave us any information about it.
So I think they're just telling us in the question stem that SGP is the un all right, so let's see. A precursor of EGP is translated from a transcript that has had one non-templated nucleotide added to the open reading frame. For adding one, we're going to mess up the reading frame, right?
Yeah.
It's telling us that it's not producing a premature stop codon, so we can rest easy knowing that it's not going to be a nonsense mutation. And then they're saying compared with the protein SGP, which is produced from the unedited transcript, so the one that doesn't have that non-templated nucleotide added, EGP most likely has the same. Okay, so we have choices that talk about the amino terminal and carboxy terminal, and then C and D are like the entire sequence. Now, if EGP has the messed up nucleotide added and SGP does not, they shouldn't have the exact same sequence. And also, they wouldn't have one additional or one fewer, right? Because you need how many nucleotides to make one amino acid?
Three.
Correct. So if they're just adding one nucleotide, that's not enough to add another or to add or remove another amino. acid, right?
I see that that makes sense. That's a good call.
Mhm.
Um,
And now it's just a matter of will they have the same amino terminal or same carboxy terminal?
mhm.
And so you want to think in a peptide, what comes first? Because we know there's going to be a C end and a amino end. Which comes first?
That's the amino end. So I feel like it will probably be A.
Mhm.
Like that makes sense. Yeah. Okay, that was like not as bad as I thought. I feel like.
Mhm.
Cool.
So basically, what we did was we read it, we saw the buzzword single-stranded negative sense RNA genome. And then they give us more information, like, used as a template, um, requires RNA-dependent RNA polymerase. And we talked about how a negative sense differs from a positive sense, and that a negative sense, virus has and what a negative sense genome is it's reflecting the template, whereas a positive sense reflects the coding strand. So if it's a negative sense virus, its template strand isn't going to be translated. It needs to transcribe it into the coding strand, and then that can be translated. So we talked about that, and then we talked about EGP and how it's proportional to EBO-V entry. And then they said that uh, acid-dependent proteases can help this EBO-V be infective. And so I wrote like 1 over pH. I wrote proteases. Like the 1 over pH thing could be a little weird. So maybe, you could have just wrote proportional to acidic proteases, but you know, they could have asked us something about like, what if we added this proton pump or something like that? And, they could have asked us something like that, but they didn't. But okay, and then we have the names of proteases like cat B and cat L, and yeah, all the proportionality stuff I wrote, simply based on the information that they're giving us. But then when we looked at figure one and we spent some time on that, we wrote out this like greater than hierarchy. Um, maybe I would even add like two of these to indicate that there's a much larger difference. It's not like cat B is a little bit more infective than cat L. It's significantly more. Yep, that just showed us that yeah, the infectivity is highest when you like, so we know from the passage infectivity goes up with cat B or cat L, but the figure showed us that it's predominantly cat B that's producing this infectivity, cat L a lot less, and combination is the highest. And we went from there. Okay.
Gotcha.
Yep.
Yeah.
So yeah, I would just look through like these questions that you got wrong and look at look more into like this topic. Maybe look more into like endosome stuff. And just like practice writing out proportionalities and uh maybe like greater than hierarchies like this for graphs and stuff. And yeah, I think you can go from there. Um, and yeah, what do you think?
I think so too. I mean, I guess yeah, I feel like I just need to spend some more time like with my practice tests, like because I feel like maybe my reviews haven't been like as in depth as they should be. So my bad, I did not mean to do that. So yeah, I think I'm gonna do that. I don't know, maybe I guess maybe I will push it back. I mean, I'm still gonna submit my application beginning of June, but I think I'll probably just I mean, because I was gonna submit with what do you call it? Without my MCAT score anyway, because I was gonna do June 14th. But I guess like June 28th isn't that big of a difference, honestly, in the long run.
Okay. So now, since I for some reason didn't tell you to, I don't remember what, but if you go to jackweston.com
Mm.
and
Peter's like listening to me.
yeah. Okay. Scroll down. See, yeah, Jack was an MCAT QBank. Click QBank, study QBank now, and that right there. Click that.
Gotcha.
And there's one for BB, one for CP, one for Psych Soc. These are just discrete questions, but they're going to expose you to everything you see, like all the content you'll see on the test. So this is what I usually have students start off with. For some reason, I'm sorry, I don't know why I didn't bring it up with you, but but this is what I usually have people start with. So yeah, this shouldn't take you too long. They're all discrete questions, but this will give you an idea of like what content stuff, you still have some gaps in, and then what content stuff you're good at, or that you already know. So yeah, I would say to work on this, maybe for like the next few days or something.
Okay.
And when you finish, review it. You could review it just by like reading the solutions, but for topics and stuff that you might need some more help in, you can use like whatever resources you like videos or MCAT books or stuff like that.
Yeah, I recently got into the video game. There's a lot of different, what do you call it? Resources. Like I've been watching like the Informing Future Doctors videos, which I think are pretty good. So
Mhm.
yeah.
Got it. All right, yeah. So yeah, you can do this and we can square away the and also like if you go to your MCAT spreadsheet that you have on the left,
Mhm.
down on the bottom tabs where it says like CP content, BB content, you can like, do like a control F or whatever the Mac equivalent or something is. And you can search like, because this is from the official AMC outline. So you can see like officially, what's fair game on the MCAT. And yeah, and then you can use the drop down menu to, adjust like your level of understanding for this stuff. So if you like, for instance, if you go to BB content, and type in the find bar, type in maybe like virus. Okay. And now let's scroll down a bit. So okay, so like virus structure, you could see like this. If you can kind of make this cell a little wider, or like you can kind of touch over here. And then yeah.
Uh, Oh my goodness. There you go.
So nucleic acid and protein and enveloped and non-enveloped, genomic content, RNA or DNA. And they probably have some more stuff on like negative sense or something. Maybe if you type in sense over here, like in the over here, sorry. Oh, not there.
Out there. Where?
Sorry, in the search, control
Oh,
F.
my bad. Put virus sense.
Oh, just sense. Missense, nonsense, codons. Okay. But yeah, I guess we don't see it exactly, but yeah, you can use this to like, yeah. So I would say do that, check Western content diagnostic, and then when you look at the ones you got wrong, you can consult this and adjust your level of understanding depending on how you do, and then your review of that stuff. Okay.
Yeah, I feel like I think I got it. I just need to put some more time in. I have I have a little bit of time. I have like a week or two to get it together, and we're gonna get it together.
Yeah, yeah, totally. So awesome. I will see you next on, I don't know if you signed up, but
No, not yet. Are you working on Monday? It's a Memorial Day.
yeah.
Okay, cool. So I'll probably book something with you on Monday then.
All right, awesome. So let's see. For today should be like 1:30.
Gotcha.
And yep, good luck on the content diagnostic, and I'll likely see you sometime on Monday, okay?
Okay.
All
All
right,
right. Have a good
you too.